Okay, so today's video is going to focus on reproduction, in particular meiosis, and how meiosis can contribute to sexual reproduction. So there are two basic types of reproduction, asexual and sexual reproduction. So over here on the left, we have asexual reproduction. Okay, this is an example of binary fission. Okay, remember, binary fission was how prokaryotic cells are able to reproduce. And with asexual reproduction, you're producing a clone. And by a clone, we mean that it is genetically identical to the parents. Okay, just like you did with mitosis. Okay, so genetically identical to the parents with asexual reproduction. With sexual reproduction, over here on the left, sexual reproduction, there are two organisms that share genetic material to create an offspring. You can see here in the top picture, there's a picture of fertilization. So there's a sperm cell and an egg cell. So the sperm cell coming from the male, the egg cell from the female, and they combine to form the zygote. And now that combined cell has DNA from both the mom and the dad. Okay? And that will grow and develop into the offspring. But the offspring then is different from the parents in sexual reproduction. It is a mix of the two parents, so it is not a genetic clone. It's genetically unique because of the combination from mother and father. Okay, let's do a little bit of review from before. So before we've talked about homologous chromosomes. Y'all hopefully know from your vocabulary now, remember that homo means the same. So homologous chromosomes were pairs of chromosomes. Remember, they code for the same thing. So, for instance, you get a set of 23, so humans have 46 chromosomes, right, 23 pairs, and you get a set of 23 chromosomes from mom and 23 chromosomes from dad, right? That's how we get our 46 chromosomes. So 23 chromosomes from mom, 23 from dad, we get the normal that we should have, which is 46, which means you get a set of everything in your body from mom, and a set that codes for everything in your body from dad. You get a set, you get information about hair color from mom and from dad. You get information about eye color from mom and from dad. You get information about what kind of hormones you're going to produce from mom and from dad. You get a full set of information from each parent. And those are going to be grouped together, remember, into these homologous chromosomes. And so homologous chromosomes are, remember, they're chromosomes that code for the same thing. So in our normal cells that have 46 chromosomes, those chromosomes are grouped into these homologous chromosomes. They're grouped into these pairs. And that's what's called a diploid cell. So in humans, for instance, a diploid cell has 46 chromosomes. You'll see these are color-coded. Okay, so the pink ones code for the same thing, the purple ones code for the same thing, and the yellow ones code for the same thing. So those are paired together in homologous chromosomes. Okay, and for instance, mom, while the other one came from dad. Okay, and so your body will pair those up. When we're going to reproduce, though, and we're going to pass on our genetic information, we've got to take this 46 that we normally have, and we've got to narrow it down to 23. Otherwise, the offspring don't have the right number of chromosomes. And when we cut that chromosome number in half, that's what's called our haploid number. So, for instance, in humans, the haploid number is 23. Let's say you were talking about an organism that had 16. The diploid number is 16. Well, then the haploid would be 8. Or if you were told that the haploid number of chromosomes was 10, then you know the diploid is 20. Okay, so in the diploid, they're grouped into their homologous pairs. In the haploid, they're all by themselves. And so if you look down here at the bottom, okay, this right here, this meiosis, this is an overview of meiosis. And meiosis is how we make the gametes. Okay, so meiosis is we're forming the gametes. And those gametes are haploid. Okay, those gamete cells are haploid. They have to have half as many chromosomes. So if you look here, there's two cell divisions that happen in meiosis. Okay, so we've got this first one happening here. So meiosis does do DNA replication, just like mitosis did. So the cell jumps up from having 46 chromosomes to having 92 chromosomes. Okay, and then in meiosis, we'll do some more things to add to variation. We'll talk about that in just a minute. But then the cells will divide 2 times. Okay, the first time, that replicated DNA splits out. So I go back to 46, 46. Okay, so for instance here I would have 46, I'd have 92, I'd go back to 46 and 46, and then in my second cell division I would split those into 23, 23, and 23. Okay, and I would end up now with my gamete cells. Okay, so I know that got a little messy, so let's just kind of make sure we're clear here for a little bit. Okay, so meiosis is going to be, like I said, a little bit different than mitosis. Okay, I still have to replicate my DNA, so that's the same. Okay, but in meiosis now, I have 2 cell divisions instead of just 1. Remember, mitosis had just 1 cell division. So I have 2 cell divisions. I end up with haploid cells, okay, or gamete cells. So I end up with cells that have half as many chromosomes, okay, due to these 2 cell divisions. Okay, and so they are genetically different than the parent cell. Okay, they are genetically different than the parent cell. Remember, mitosis, they were genetically identical. Mitosis was done to replace skin cells. So if I have 46 chromosomes, I still want 46 chromosomes. Meiosis is done for reproduction. So these cells end up being genetically different than the parent cell. So meiosis occurs in both males and females. In females, it's called oogenesis. Hopefully from our vocabulary, we can sort out some of what this word means. Oo is egg, gen is to produce. So in females, it's oogenesis, and it's egg production. Okay, in females, remember how we looking at that overview of meiosis, we saw that it made 4 new cells. And in females, because they're producing the egg, and the egg has to provide all of the nutrition for that growing and developing embryo, they produce 1 viable egg. Viable means that it's good, it will survive, it's able to reproduce. So 1 viable egg and 3 what are called polar bodies. And these polar bodies are not used for reproduction. They're reabsorbed, they are not used for reproduction. In the females, in the males, we're producing sperm cells, so it's spermatogenesis. Again, we've got gen in there to produce, so they're producing or making sperm cells. Okay, so spermatogenesis produces, it occurs in the testes of the males, and it produces 4 viable cells or gametes every single time. Okay, so males, every time they go through meiosis, produce 4 viable, usable sperm cells. Females, every single time the ovaries go through oogenesis, only produce 1 viable egg. The other 3 polar bodies are, like I said before, they're reabsorbed. So what's the point in doing meiosis? Okay, and so the point in doing meiosis is that it's going to increase our genetic variation. Okay, and that is extremely important, especially when you get into evolution. If there is no genetic variation, then there is no ability to have or certain organisms better suited to the environment because there's no differences in traits. If a disease came through and everyone was susceptible to it, if there was no genetic variation, there would be no chance that somebody would be resistant to this disease. So genetic variation is very important to us, and that's the role of meiosis. It absolutely does not make new genes. Okay? It's not making new genes. It's increasing the variety with the genes we have because it recombines the genes. Okay? It recombines the genes, both in the sense from mom to dad, okay, combining the genes from mom and the genes from dad into you. So that's a recombination of the genes, so we get variety there. And then in forming the egg and the sperm and going through the spermatogenesis and oogenesis, the meiosis will also help increase genetic variation there as well. Okay, and there's 3 ways it can do that. Through what is called crossing over. and we'll look at that one a little bit more in depth in just a minute. Okay, independent assortment of the chromosomes, as well as random fertilization. Okay, and we'll look at all three of these to get a little bit better understanding of this. But again, that's one of the main things we need to grasp here, is that the point of meiosis is to increase genetic variation by making new combinations of genes, not brand new genes. So let's look at how the egg and the sperm cells become different. Okay? Not just the recombination that's formed when they come together, but how they get 4 different sperm and egg cells. So if you look at up in the right-hand corner here, we've got our overview of meiosis again. Okay, so here's the DNA replication has occurred, the first cell division, and then the second cell division. Remember that second cell division is producing the 4 gametes. So let's talk about sperm cells, because we produce 4 different viable gametes with sperm cells. And so you'll notice these sperm cells have half as many as this. So the original cell had 2 pairs of homologous chromosomes. So its diploid number was 4. Okay, that was its diploid number. 2 pairs of homologous chromosomes. They got one from their mom and one from their dad. One from their mom, one from their dad. So now they're going to take those homologous chromosomes, and again, that's the point of meiosis, is to split those up. Okay, but if you come look here at the end, okay, these new cells are haploid. Their haploid number would be 2. They have the half as many chromosomes as the original cell. And you'll notice these chromosomes are a mix of the gray and the red. Okay, and they're a mix of the gray and the red due to crossing over. And so crossing over gives more genetic variation. You'll notice here as you look at these are 4 different cells as opposed to being 4 copies, 4 haploid copies. They're 4 totally different haploid cells. That's how if you have siblings, especially if they're the same gender, that you guys don't look exactly the same, because all the sperm cells and all the egg cells that are made are not exactly the same. And so crossing over happens during meiosis, and what crossing over is essentially the homologous chromosomes trade parts. And they're going to trade homologous parts. Remember, homologous chromosomes trade code for the same thing. Okay, so homologous chromosomes code for the same thing. So these homologous chromosomes, maybe that word would help here. These homologous chromosomes trade homologous parts or areas. Okay, so for instance here, if we're looking at the beginning here with A and B and the purple and the turquoise chromosomes here, okay, these are homologous chromosomes right here. So they code for the same thing. So if I switch out parts of these chromosomes, it doesn't matter. It's not like I switched out something that codes for hair with something that codes for digestive enzymes. Okay, I've switched out things that code for the same thing. And so this happens, you'll notice it happened up here during the first cell division here, before that first cell division takes place. So this crossing over just basically allows homologous parts of those chromosomes to switch places. And so now I added more variety to my gene pool. I've made extra combinations of genes. And again, that's the whole point of meiosis, is to add genetic variation. So let me show you one more picture to help just really kind of hammer home this crossing over and exactly how it works and what's happening. Okay. So in case you were struggling a little bit with the other picture. Oh, me, oh my. Okay. So maybe we won't show you one more picture. Okay, so here's the picture from the jumbled mess just a second ago. So just to shows you just that extra step here with crossing over. Okay, so I've got my homologous chromosomes. They replicate here. Crossing over happens right there. So you see those things code for the same thing. You've got allele A, you've got gene A, gene B, gene C. So we're just crossing over here at gene C. So now I've got a variety of chromosomes that can go into my individual gametes so that all my gametes are different. So independent assortment is another way that meiosis can help increase the variation. So you'll notice your homologous chromosomes here, they split down the middle. Okay, and so as they split down the middle, That's how you end up with your gamete cells. And so when they split, there is no set rule as to where they all need to go. For instance, if they are lined up like this in metaphase, okay, there is no set rule that because this one went this way, this pink one also has to go this way. Okay, this pink one could go this way, so this pink one went this way. They don't care what the other pairs of chromosomes are doing. Remember, you've got 23 pairs of chromosomes in your body that are going to be separating out. Pair number 2 does not care which direction, how pair number 15 wants to split apart. And so that's what it means by independent assortment. Okay? When these pairs split apart, they are not influenced by one another. The last way that meiosis can help increase variation is through random fertilization. And this is technically not during meiosis, okay, but it's during reproduction in general. And that's just the fact that any sperm can fertilize any egg. As long as the proteins can penetrate the egg, any sperm can fertilize any egg. It is not set that this one sperm from this one person has to fertilize this one egg from this one person. Okay, but crossing over and independent assortment both happen during meiosis, and they both are helping to increase the variation. Okay, so let's talk a little bit now then if something went wrong. Okay, so y'all hopefully remember some of this from mutations. But remember when we were talking about mutations, and we talked about them being able to be passed on. And so these are all examples of chromosomal mutations. And these are mutations that if they are in the gametes, they can be passed on. But remember, a mutation that does not occur in the gamete cells cannot be passed on to the offspring. It has to occur in those gamete cells. Remember, there's examples of the inversion, the duplications, the insertion and deletions, as well as the translocation between those non-homologous chromosomes. Again, all of these were talked about in mutations. But just a reminder that these are going to happen, they're going to happen most commonly during crossing over. When those homologous chromosomes are trying to switch parts, okay, that's when you run the biggest risk for one of these chromosomal mutations to happen. And so these are both examples of non-disjunctions. Remember, those were a type of chromosomal mutation. Non-disjunction is when the chromosomes don't separate properly. So instead of having one from mom and one from dad, you may end up with a triplet, which would be our trisomy. So that means a pair did not separate properly, and you either got 2 from mom and 2 from dad, and then you got 1 from the other parent. Okay, so they didn't separate properly, or you end up with what's called a monosomy, where they didn't separate properly, and so you're short a chromosome. Okay, one of the gametes did not have a particular chromosome. Instead of having an extra, it didn't have one at all. So non-disjunctions are when the chromosomes don't separate properly. Again, our common ones are monosomies and trisomies. And you can see these on a karyotype. So let's talk for a minute about karyotypes, and then we'll be all set. Y'all remember from mutations, karyotypes are just pictures of the chromosomes. So for instance, this up here is a typical karyotype. There's no monosomies, there are no trisomies, everything's just paired up. And so if you look at how they're paired up, you can start to figure out how the karyotype is made. So the first thing that needs to happen is the DNA needs to be stained. So we stain the DNA so it shows up with these banding patterns that you see here. So we stain the DNA, you get the banding patterns, and once based on the banding patterns, you can group them into homologous pairs. And that's how the karyotype is arranged. They're arranged into their homologous pairs that code for the same thing. And you can determine homologous pairs by the banding patterns. And then once they're grouped into homologous pairs, they're then arranged from largest to smallest. The first 22 of them are. So there's my biggest one. There's my smallest one. And those first 22 are called autosomes. Okay, so they're arranged largest to smallest. 1 through 22 are our autosomes. Autosomes are coding for everything in your body essentially but gender. Okay, so 1 through 22, they're stained. We match them up homologous pairs. Okay, and then we line them up tallest to shortest. And then the last pair, pair 23, those are the sex chromosomes. And so those may not always be a pair. For instance, if it's a male and it's an XY, X and Y don't match. Okay, so they're a pair, but they're not a matching pair. In females, they're a matching pair because you're going to have two X's. But in males, you may have a large one and a short one because you have the X and the Y. When you look at this picture over here, okay, let's take this one and get it a little bigger here. Okay, so if you're looking at this one here, you can see how they've stained these chromosomes over here on the left, and then they've been matched up into the homologous pairs. This right here is just a drawing, so you can see the banding pattern a little bit better. But both of these then have this same banding pattern, so they're homologous pairs. We're paired up tallest to shortest. Okay, so those are my autosomes. And then at the end, I would have the pair of sex chromosomes, pair 23. Okay, so hopefully we should be all set with meiosis, and we'll be doing this right when we get back from the break.